Hi, my name is Noemi Soto, and today my presentation is going to be about fungal farming and how ambrosia beetles choose the best plant hosts to cultivate their fungal chambers. And I've been in the University of Florida as a student for about a couple of years, and I'm currently in my second year as a master's student for the Department of Entomology and Nematology. And as I like to begin, I thank you for watching this presentation. Ambrosia beetles are mycophagous insects, and their primary, as meaning that their primary food source is fungi. And through mutualistic associations with fungal symbionts, their coevolution with these fungal symbionts has made them fungal farmers. Therefore, they tend to make fungal gardens within trees. Now, as they are social insects, they are very, very highly selective on the fungal symbionts that they choose. And they will pass down these highly selected fungal symbionts to their progeny via microangia. And they will surround their brood with these fungal symbionts. Common fungal symbionts are Ambrosiella and Raffaella species. Ambrosia beetles screen for environments that are optimal for fungal growth. And this is mainly due to the fact that they want to discourage the growth of invasive fungi and competitive fungi. And such examples are the Aspergillus and Penicillium. Trees that are healthy are not attractive to ambrosia beetles because they lack the product ethanol. Ethanol is very important for the selection process for ambrosia beetles because of the fact that if a tree is emitting ethanol, it is a signature sign saying that the tree is decaying. And also, it is a great important factor in the selection process because ethanol is not only an attractant, ethanol is also an antimicrobial agent, which allows it to discourage the growth of these invasive pathogens. Now, when an ambrosia beetle is successful in finding a decaying tree, you'll notice on the picture on the right of these tooth-like, hair-like structures on the outside of the bark. This is known as frass, and this is an indication that there are tunnels being built in this decaying tree. There was a study conducted by Ranger et al. As the title listed to the right, they studied the plant host selection behavior in a species of ambrosia beetle known as the Xylosandrus germanus. As ethanol is an antimicrobial agent and an attractant, therefore, this species of ambrosia beetle is reliant due to the fact that it will produce successful progeny and it will be considered an optimal environment for mutualistic fungal cultivars. So, within this paper, the approach to whether or not they can detect whether the ambrosia beetle species is using ethanol more than just an indicator of whether a tree is dying or not, they used dogwood and redbud trees in a field tested study and irrigated them with levels of ethanol ranging from 0 to 5%. And they measured the number of ambrosia beetles of this species, the number of attacks every tree had, and they followed the progression as they counted the quantity of successful brood and fungal chambers in each of the trees. In their second approach, they also did a fungal bioassay within the lab as they cultivated pathogenic and mutualistic symbionts and measured their growth at variable concentrations of ethanol. And here are the results given by the authors of the paper, and the results on the left are given by the first approach. The first approach shows here the comparison of results of the dogwood and redbud trees. As clearly they have similar results, showing that ambrosia beetles do not select based off plant host species. Also, they've also compared ethanol baited trees and ethanol irrigated trees, and there happens to be no gallery nor eggs or larvae within these trees in both cases. This is also a clear distinction that they're choosing trees that are ethanol irrigated because of the fact that it's an antimicrobial agent. It's not just an attractant. And there's also a positive correlation in certain categories where the higher the ethanol percentage of irrigation in the tree, then there's also a higher amount of tax, a higher amount of foundresses, and it will vary when it comes to fungal chambers, eggs, and larvae. But compared to when they are baited, the fact that they are present is also a good indicator for supportive evidence of their hypothesis. The results on the right were from the second approach of the paper, where they measured the amount of growth from the fungal symbionts that they grew in increasing percentages of ethanol in the media. The first figure is a species of Ambrosiella that is highly associated with Xylosandrus germanus. The figures here indicate that there's high correlation, high positive correlation, as the density of this species increases at ethanol in the media. This is an indicator that this species is thriving off of the ethanol, as it can detoxify the ethanol within the media. Compared to the figure below, there are other species here, but here it's only indicating the dry weight. But they're comparing the fact that Ambrosiella species and Ramphiella species, which are mutualistic fungal symbionts to ambrosia beetles, are positively correlated with ethanol in the media, to which they reach a limit. But these are not associated mainly with Xylosandrus germanus, but they're representing the fact that mutualistic fungal symbionts compared to competitive fungal symbionts listed below, Penicillium and Aspergillus, there's a clear indication of which fungal symbiont can detoxify the ethanol. Clearly, the ones below can't. Therefore, this is why it shows that ambrosia beetles are highly selective by choosing trees that are high within ethanol so they can therefore breed a specific species of fungal symbionts. In conclusion, the authors have been able to support their hypothesis that ethanol serves more than one role as being an attractant, but rather a good, reliable source of fungal chamber maintenance and a good source to find areas, environments that allow for them to brood successful progeny. And the results have shown that trees that are irrigated with ethanol have quantities of progeny compared to trees that have been baited with ethanol where there are no progeny present. Further supports their hypothesis that ethanol is more than just an attractant. Also, one of the most important conclusions derived from the paper was that they were able to deduce that fungal symbionts of this species of ambrosia beetle are able to detoxify ethanol. And this is also great because those that cannot are considered less competition. And this is evidence of coevolution due to the fact that ambrosia beetles will select fungal symbionts that can live within ethanol mediated environments that disallow for pathogens. And this concludes my presentation, and thank you for listening.